just to remind our growers, caregivers, that we already have severe and impactful it's called Candidates Liberibacter causes associated with citrus greening, and it's devastating our industry. And we don't want anything else in terms of diseases on top of this. So what I wanted to remind you that there are a couple things that we can do to ease the HLB-associated problems. And then what we should be looking for to prevent other diseases to exacerbate the impact of HLB. And I'm trying to advance my slides. Okay. And I'm assuming that after 17, 18 years of talking on HLB, living and breathing HLB in the groves, you are all familiar with many aspects of the HLB. So just to remind you that this is phloem-limited bacterium-caused disease that is mainly transmitted with insect vector called Asian citrus psyllid. And I'm sure you are familiar with the insects, adults, and the nymphs stages. Over here, my cursor is visible, I hope. And it causes many disease symptoms. Particularly, the characteristic symptom is asymmetric blotchy mottling on leaves, and then reducing the fruit quality and yield. Most of the fruits are lobe-shaded, smaller in size, and taste also altered because of the disease. But the most devastating effect of the HLB is, of course, a pre-harvest fruit drop. So because of this, HLB has really been devastating our industry since its occurrence in Florida in 2005. Now, HLB is not a new disease. Some of you probably will not be surprised. This is 100 years old, a century years old disease. And it's originating from the East Asia, where citrus is originated. But over there, many of the integrated pest management strategies somehow control this disease and allowed producers to produce reasonable citrus and get along with it. So, of course, we are all frustrated that many of the attempts to control this bacterium or disease in the grove failed or didn't produce enough to increase the production in our groves. So I wanted to remind you a couple of things in terms of how we, as a plant pathologist, take care of the general plant diseases and what has been done for HLB and what it worked and what it didn't work, as well as what has been done for controlling its insect vector, Asian psyllid. So as you can see on the top of this slides, under the general plant disease control, it's literally 101. Intro to the plant diseases. There are three main things that implies almost all diseases. One, especially those that transmitted with insect vector, you control the insect vector population. Second, you reduce inoculum. Reducing inoculum is removing infected material and producing next future crops in clean, indoors, out of free of disease and insects, and start again with the clean material. So these three things applies almost all plant diseases, but did not apply much for HLB because when HLB was found in Florida, one of the main critical part of this plant disease control was the removing infected material, which we totally failed in Florida. And the reason was more than 95% of the citrus trees were already infected when we realized that HLB is here in Florida and spread all over, okay? However, over 17 years, research has not stopped, and many people tried so many different ideas and products to control the HLB, which are, some of them are listed here in red. Red also indicates that none worked well or yet to be seen that is working well, okay? So as you can see, there is no black font in my list under the HLB control. So none of the control strategies so far being helpful with the controlling the HLB. But one particular line that I underlined and I said, remember, we also tried spraying bactericides and many other products that completely fail. So please do not do the same mistakes. And as you can see under the psyllid vector control, all are in black font, means all these management practices are working and effective for ACP. So still we can control our vectors to reduce the impact of HLB. And take-home messages, there is no single effective control strategy to control this disease, okay? Still, we don't have anything silver bullet type of single management strategies that will work for HLB control. Of course, we are not giving up on coming up with the other strategies to control HLB soon and in the future. Some of the projects that I'm involved, I just wanted to give you a little intro. For example, one project that I'm part of, working with some other researchers in Gainesville and other places, other institutions. We are even looking at the CRISPR-Cas gene editing, producing transgenic plants or producing non-transgenic plants to combat this HLB. It's one NIFA awarded project that is been going. And we do have very encouraging results. And other listed under the second title, it's the one that CRDF with the Bayer in collaboration with my team, looking for and screening different chemicals and materials that one of the largest pharmaceutical companies in the world, or agrochemical companies named Bayer, Bayer's library, to see whether they have any effective material that we can establish and show that it can be used in near future by our growers to control HLB, ACP, or in other diseases. So, and these days, as you may all aware of that, growers and industry in general excited about the permit that a company established for injecting oxytetracycline through the trunk of the citrus trees. So one of our projects, actually, with a number of other faculty here in SWFREC and in other places, we are trying to automate this trunk injection so that our growers may use to inject oxytetracycline or other potential effective therapeutics to control HLB and other diseases. So this is an ongoing project that simply it will be like a robotic arm that it will be attached on any farm vehicle that will be extended from the vehicle by the operator and grab the citrus trunk and inject the therapeutic material that we think that it will have some effect on HLB and other diseases. So this is really a hot subject in our team, for our team as well, and we are working continuously to finalize the final prototypes so that we can go out and show you soon how it's going to work and what can be done with this equipment. And another project that I'm involved also, looking at the some of the peptides that other citrus relatives that having and showing some tolerance to HLB, namely here, Australian finger lime. Probably you heard that it's been identified as a tolerant or resistant to HLB disease. And one of the peptides that produced by this tree is identified not long ago. So what we are trying to do is trying to transfer that the small peptide. Peptide is the small parts of the larger proteins that is produced in this plant to our Valencia or rootstocks that we are commonly using here in Florida. So the main idea is using the tolerant or resistant as an interstock, as you can see here, that is grafted on our regular rootstock as an interstock, and on top of that, grafting Valencia. With this, we are hoping that a peptide or any other molecules that might giving resistance or tolerance to this finger lime might move up and down to give some protection. And this is also an ongoing experiment, but soon, hopefully, we will have some results that I can share with you. So how are we going to manage HLB? I said none of the strategies that work so far. But as in the past, integrated pest management that is targeting multiple facets of the disease triangle will be most probably effective reducing the severity of HLB. So still that we have to go out and take care of the other disease and the insect vectors and control them to not let exacerbate the HLB's problem. And now we are also thinking that reducing inoculum might be possible, perhaps by injecting oxytetracycline, but this is yet to be seen because this is a really new strategy that been put in test in the grows, because in the greenhouse setting, we always had a great results showing that oxytetracycline is really helping the infected plants to reduce the inoculum and show some improvement in yield and health. And in small scale field experiments also, we demonstrated that oxytetracycline has the possibility to help plants to recover from the severe disease symptoms. But definitely, it will require additional management strategies to control or help trees to recover from HLB. And one of them is definitely will be continuing to take care of the ACPs. Because please remember, you here in person and those listening on Zoom, bacterium CLS or HLB disease agent, whatever you call it, is multiplying inside the guts of the ACP. So if you are not controlling the ACP, every insect that will visit your clean or infected trees will inject more bacterium, even though if you may inject it oxytetracycline to reduce the inoculum, ACPs will bring more and more bacterium. So that will diminish the effect or potential of OTC. So please remember that. If you forget everything else I said, remember OTC injection itself will not be enough. You have to take care of the psyllid. And obviously, in my future subject, this second part of the presentation, I will be talking about other disease, Phytophthora, that is mainly infecting citrus trees through the roots. It's a soil-borne pathogen. So in this case, I would like to remind you that HLB also infects the roots of the citrus plants. So in combination, it can be devastating as well. So please pay attention to root health while you are taking care of the psyllid population and reducing the inoculum of HLB by paying attention to irrigation and fertilization requirement of your growth. So, of course, we still have a lot of questions about HLB. What will be the perfect combination of the integrated pest management strategies? But we are pleased to see that OTC is a part of the integrated pest management, and it is not a silver bullet that will, stop the HLB problem in Florida. So every time with our teams and others' efforts, we are learning a little more. For example, you may be hearing these days how important to provide adequate nutrition and using gibberellic acid for controlling the HLB. And we quickly learned that bactericide sprays were not effective, so we stopped doing that. But now we are hoping that injecting bactericide through the trunk might provide some more adequate control for this bacterium. And we know that ACP control is working, and we should continue to control ACPs in our grows. Still, we don't know how often we can inject OTC, how much and when, and in what trees will work, what not. So we have to not let our guards down, hoping that OTC will solve everything. And ultimately, IPM suggests that we will need, at some point, tolerant or resistant varieties to combat this disease. Okay? So the second part of my presentation is about Phytophthora. Many of you are familiar with this disease. It's not a disease, it's a new disease, and it's been impacting the citrus production around the world. And it's a soil-borne pathogen that can infect roots, trunks of trees. And there is one type of Phytophthora species that can jump from the soil by irrigation water or rain to the fruits and infects the fruit that causing brown rot, as you can see here. And this disease is still causing about 5% of yield reduction. And 5%, I think, is quite a bit, especially when you are suffering already with HLB. So this is another disease that our growers must continue to be on top of it and take care of it. So I gathered some of the Phytophthora incidence data from Syngenta that's tracking the number of propagules. Propagules are the type of formation of this fungus that lives in the soil, and when the optimum condition comes, that grow and infects other plant material. So this company's sampling demonstrated that over the years, number of propagules, those units of the fungus that can cause disease, increasing in the soil. And because of the HLB, as you may know, it's infecting the roots, reduce the root number in tree, together is becoming more severe, causing Phytophthora to exacerbate the symptoms of HLB and making the already bad situation worse. So another set of data also suggests that some of the propagules stays some longer time in soil from previous years when we are not taking care of this pathogen and accumulating. And as you can see here, for instance, Southwest Florida's bar here with the yellow one, the number of propagules per gram of root mass that were examined, it's really, really high. And establish protocols to control the Phytophthora in citrus groves. As I said, it's not a new disease, and this is a disease that before the HLB era. And production guides suggest what is written here. So when you have tested your soil and number of propagule per cubic centimeter is more than 10 or 20, suggests you to use some chemical intervention to suppress the propagule and reduce the inoculum in the soil so that you can protect your roots. Again, these applications are protective, so don't forget, you have to apply these chemicals before it's too late. So, and Phytophthora is, it's not a fungus anymore, it's the oomycetes, and it's mean fungus or animal that can swim and come to your roots by swimming and infect your roots if you do not protect, especially fibrous roots. That is also increased by the Silas infection. So one key interaction here that the Silas infected trees are producing more fibrous roots, and it's unsynchronized way. So it's mean plant produce most of the time food or infection material for Phytophthora. So that's why actually some of the recommendations made here might not be effective anymore. So we have to remember that citrus plants are now producing flushes, leaf flushes and root flushes out of sync. So there will be always material from above the canopy for ACP to feed and inoculate more. Under the soil, in the root, more fibrous roots that is attracting, because fibrous roots are exudating chemicals or substances that attracts the Phytophthora to take an action and swim towards the roots and inoculate. Okay? So again, and this propagule counts that you can definitely do in your groves, and I think the Syngenta, Matt, correct me if Syngenta is providing this service free of charge if you are suspecting Phytophthora in your grow. So, and if you have results showing that more than 10 or 20 propagule per cubic centimeter present in your grow, it means you should definitely take action. And I just put a reminder for myself to tell you that this threshold, 10 to 20 propagule, is actually established 20 years ago. So it's mean this was established before the HLB era. So I don't know if this 20 propagule is, if it's not too late to start taking care of the Phytophthora. So as I was trying to suggest that the Silas infection is changing the hormonal balance of the infected citrus trees, causing flushing all the time, shoot flushes and root flushes. So that's making these products, the effectiveness, little in the gray area, because plant is not responding well anymore because it's infected with HLB and a number of other physiological and immunological things are out of sync and not working. Plant is not responding your chemicals that you are putting for Phytophthora, for example. Okay, and As I was trying to show you from the data that I gathered from the Syngenta's report, that the propagule counts are increasing in the soil, and the root mass that are citrus is decreasing. So it's mean rest root available for more fungal spores to inoculate. This all in combination suggests that you should definitely make some adjustment to your Phytophthora management strategies. For example, definitely you have to improve timing of your application, thinking of flushing of leaves and the roots are unsynchronized, and number of the application may be increased, or the dose of the application. So we have to look at some sort of adjustment. Basically, what I'm saying, don't do whatever all of the years in prior years that you've been doing to control Phytophthora. So this is the time that you have to have a closer look to change some practices to make the chemicals that are available these days effective to control the Phytophthora. So just to summarize, if you have a 20 or more propagule per cubic centimeter of soil, and you have HLB infection in the grow, I bet you all have, this is the time to change management. Okay, and remember, Silas and Phytophthora, as I also put some reminder for you that especially Dr. Jim Graham's group been looking at the root health and Phytophthora and Silas interaction over the years. They already suggested quite a bit of evidence that roots are impacted more than we thought by the HLB alone. So we should not let Phytophthora to exacerbate the situation. And again, treatments are protective, so apply at or just before root flushes, and especially if you are expecting rainy days to come. By the way, I'm not sure how I'm doing with time. Mongi, can you track that and give me a heads up? Thank you. So again, since you cannot just go and protect every flushes that plant gives as a root flush or leaf flush. By the way, I just put this scheme here to remind you that after every shoot flush, there is a root flush coming that we don't see it, right? So please remember that. After every shoot flush, there will be probably some root flush coming. And again, since Silas disrupted the shoot and root flush cycles, this is not the case anymore. So you have to make a decision here, what flush to protect, what shoot flush to protect, and what root flush to protect. So we recommend here that you have to identify the root flushes that you think will give you the best return if you protect them. Okay? So I put some cloud or thinking icon just to give a little bit of my thought. And I'm sure that some growers are preparing themselves to use oxytetracycline injection right after the harvest, which is just before the spring flush. That indicates here flowering and fruit stage around March, April time. If you are planning to inject oxytetracycline hopefully will reduce the inoculum in the tree. And if you continue to take care of the ACP population to protect the next big flushing cycle, you will be making a very wise investment to start the production here. And I am thinking that maybe you will not need to protect the first root flush that after the spring cycle, but just before the rainy season starts, then you have to really think seriously how to protect your root flushes from Phytophthora infection. So this is suggesting that before the rainy season, around June, July, you have to think of taking care of the Phytophthora. And if you can make this arrangement, I think you will be more successful reducing the impact of these diseases on your production, and hopefully you will have some return by having a good quality fruit or less fruit drop that you can sell your fruit. Third and fourth part of my talk. Yes, it's a heads up, it's a 4-part. Remaining 2 parts are 2 new diseases that emerge in other parts of the world where citrus is produced. And thank God that we don't have these diseases here. I'm giving just some introductory presentation so your eyes can get used to the symptom of this disease, and you can think for the future to prevent bringing this disease on top of the HLB and Phytophthora problem that we have. Okay, so first disease that I will be talking about is called citrus leprosis virus. Again, it's not a new disease, and as a matter of fact, used to be here in Florida in 1860s, but it just disappeared. This disease disappeared in 1960s, and presumed to be because of the freezes that killed most of the citrus trees. And back then, the use of sulfur to take care of some arthropods in citrus groves reduced or eliminated this disease. So we don't have in Florida. Yet, leprosis is considered one of the most important emerging citrus diseases in the world. And unfortunately, it's spreading and causing economic losses in southern border and many countries in the South America. So we have to be vigilant and look for these symptoms. And if we see such symptoms or suspecting disease in sort, that we have to take action. Just to remind you that citrus leprosis is a non-systemic virus, means it infects only tissue where it's introduced. So it's not moving like HLB or citrus tristeza virus from where infected. It doesn't move anywhere. And it's called localized virus. And you will soon see and understand why we are getting chlorotic or necrotic spots. It's not everywhere in the tree or on the leaf. It's indicating that localized presence of this virus. And there are 12 or more different viruses as causing, we can call the complex of group of viruses causing similar disease symptoms. And we will call all leprosis virus for practicality. And this virus, as I was trying to say, these viruses are separated, N type and C type. This is a little more details, but you don't have to worry about it. It comes just because depending on when virus is replicating in infected cells, it's giving us plant virologists to distinguish between viruses that causing leprosis. Most important thing to remember here, though, this virus is transmitted by Bravipalpus or flat mites. Okay? Virus cannot move, but mites can do. Okay? So remember, these are mite-transmitted viruses. So if you have a flat mite, please pay attention to some of the symptoms that I'm going to show. As a matter of fact, we are in need of identifying some groves here around Immokalee where you suspect or you have flat mites, that so we can collect and identify them to see whether these mites are the species that can acquire and transmit this virus. So please get in touch with me if you are suspecting any flat mite occurrence in your groves these days. So this is a virus disease that do not transmit by seeds, and it's moderately can be transmitted by grafts. So in other words, if you take infected tissue and graft it, it will be transmitted, but usually it will not. So the most important or main vector for transmission of this disease is flat mites. And here is one of the electron microscopy photograph of the mites. And I will start showing the symptoms of this disease, and hopefully it will make your eyes to get used to it. So when you are scouting your groves, you may come up, you may see such symptoms. So please keep in mind that leprosis is a virus disease that we don't want it to re-establish here in Florida. We have enough problems. So I was telling localized lesions on leaves, fruits, and stems. So these are the places where the green tissue are being fed by the mites. Remember, mites goes to the well-hidden green flash, nice material to feed on. And if they find such place, they will not move. And during the feeding, they will introduce this virus. Thus will cause localized symptoms. However, if you don't take care of mites or this disease, it will, of course, like other diseases, will lower fruit quality, and it will cause premature fruit and leaf drop, and ultimately will impact your yield too. Okay. Back then, this disease was called scallybug, and you may understand why it was called by looking at this picture. Now, most scary part of this picture is look like what? Anybody? Are you familiar with these symptoms? Canker. Citrus canker cause almost identical symptoms on young twigs and branch. So please look for other symptoms as well. So only looking at this branch symptoms will not give any indication if you are looking at the leprosis or some canker. So one of the most characteristic symptoms of leprosis is, as I was saying, localized symptoms that you can see here on the leaf, chlorotic spots, and there is one necrotic spot right in the middle. This is the necrosis caused by feeding, mite feeding damage. And the rest is where the mite is introducing virus and replicating, hence the localized chlorotic rings or spots, okay? Again, on the leaves, both from the upper side or the lower side, you will see the same chlorotic spot. Usually it will be single spot like this, but depending on the flat mite population, many mites can be feeding on the same leaves, and these spots can coalesce and become a blotchy model or blotchy appearance. And as you can see on the right side, for example, okay? But I think when you look close, you will be still able to see some sort of rounded and concentric rings type of symptoms on the leaves. And here is some more example. And sometimes also the older lesion especially will develop some gummy rings, as you can see here from the above and underneath of the leaves. It's both visible, okay? In early stages, It's very chlorotic, and later on develop some necrosis, means that tissue are dying in the infected areas. So here's another chlorotic lesions, spots on the leaf on citrus plants. And as I was saying, different type of the viruses causing very similar symptoms. So these symptoms are similar, but somehow little different. For example, is the size of the lesions are smaller, and necrosis is maybe in the middle is bigger than what I have previously showed. Nevertheless, both are caused by the leprosis virus. Okay, so again, these are the symptoms on the twigs and branches. So early, it develops that rusty necrosis, slightly raised, corky apparent tissue, and it can be easily confused with citrus canker. So please look at the other disease symptoms and the vector presence. So probably you will not see this disease symptoms on the young branches if flat mite population is not so high. But nevertheless, worth looking everything. Some more symptoms for your eyes to get used to it. These are all caused by citrus leprosis during the flat mite feeding on a green tissue. Okay. Here are the lesions on the fruit. So early stages. I'm trying to demonstrate in a cartoon mode here. You will see that the green center, but the bright yellow halo around it. And later it will become, as the fruit is maturing and getting older, the symptoms also will change, and it will become more necrotic in the middle, but yellow halo will remain. So this can be also be confused with the black spot or canker, but the difference here is these lesions will be very smooth. So when you touch the surface of the infected tissue, you will not feel any bumpiness, but sometimes very late in the advanced form, those lesions might be totally necrotic and become, sunken, as you can see here in the last picture over here. Okay, but usually it will be very flat and smooth. So some more symptoms on the fruit. Again, necrotic lesion in the middle where the feeding occurred, and yellow halo around it, and very smooth symptoms on the citrus fruit. Again, it's you have to be careful not to confuse with the black spot. Those who are not trained for recognizing black spot, it's a subject that Megan Dudney after me will be talking about. So you may make an association how these symptoms looking and how Megan's pictures will look like, okay? But remember this, we are here, always we are here for you. If you need help with diagnosis, please reach out to Megan or me or any of the your citrus agents. We will be happy, more than happy to help you. So here are some of the pictures that I thought is very easily can be confused with the leprosis and canker. As I was saying, citrus leprosis over here is more smooth and flat compared to citrus canker on the leaves, a little bumpy, and erupted necrotic tissue is very visible, and you can feel with your finger. The same thing is on the fruits, as you can see here, flat necrotic and yellow halo, whereas eruptant and very rough surface on infected fruits. But it's very similar on the twigs. Okay, so please remember. So again, leprosis was once here in Florida, means can come again, and our climatic conditions are very conducive to reestablish this virus. So the most important thing to remember here, not to bring this virus back to Florida. please, that's why, do not bring any planting material from outside of Florida that may have a vector like mite that you can definitely not see with your eyes. And it can be a virus in the plant that it doesn't show yet symptoms. And my last part, you will see that this new virus sometimes do not show even symptoms. So how are you going to recognize the material that you are bringing do not have this virus? Okay, so the most important thing to remember, please, do not bring any material from outside of Florida. And I just wanted to remind you that preventing this virus to come to Florida is much easier than trying to control after establishment. So please do not bring any material. And again, we are all here. You have faculty to serve you and help you with the diagnostics and control measures if happens that you see some suspicious samples that you think that leprosis might be in your growth. So this is the last part, and it's the viral disease that we do not have in Florida. Thank God, we do not have. Okay, so we do not have this virus yet, but it's a virus that you will see soon that it's even more than CTV. That can be transmitted by a number of different ways. And this is an RNA virus that can infect many citrus cultivars and hybrids, and sometimes show symptoms, but sometimes do not. And this is different than leprosis. This is a systemic virus, meaning it can move all parts of the plant and may or may not show symptoms. So just a little bit background about this disease. It's first found in Pakistan in 1988. And since then, it's been spreading and emerging important citrus virus that causing economic losses in mainly Asian countries that producing citrus. So far, we didn't have we didn't have. Now we have. We did not have this in Americas, but as of 2022, there were incidences in California where this virus was found in some of the dooryard citrus. And now we have to be vigilant and look for this virus and to see if we have it. And if we have it, we have to eradicate this virus before it gets out of control and cause more trouble. So let's look at some of the symptoms. The virus name is citrus yellow vein clearing virus, or we will say CYVCV. It's a mouthful, or I may say yellow vein clearing, so it will be much clearer. And the name is coming from actually a symptom. It's causing yellow vein, as you can see here in this lemon trees. Very distinctive yellow veins, and almost like veins are cleared. That's why it's called citrus. First, it was found in citrus, yellow vein clearing, and the agent is virus. Okay? So this virus, especially causing damages on lemon, on sour oranges, and other cultivars that I will probably show more pictures that you can remember. So again, the symptom is very depending on what you are growing, where you are growing, and how you are growing. So it's a little confusing, okay? So you will see that these symptoms are very distinctive and it's visible, but when the leaves are maturing, they might go away. Or if the temperatures are high, the symptoms might go away. But still, virus is there and impacting your yield. So that's why you have to worry about this virus. And again, it's spreading rapidly and symptomless in most citrus cultivars, causing considerable losses. And it's not only infecting on citrus, it's infecting citrus species cultivars, hybrids, and relatives. And it was found in Turkey, for example, can infect grapes. And other herbaceous hosts are listed here, bean, cowpea, and quinoa, for example, and some weeds that you may have. Okay, so this is actually it has quite a bit of host range. So there are multiple hosts exist in your or around your grow. So we have to be vigilant. And here are some more symptoms that you may remember when you are walking your grows. Again, most distinctive symptom of this virus is yellow veins or clear appearance of the veins, and some wavy appearance of the leaves and deformed leaves. And in some cases, this water-soaked appearance, it's more clear here, not here, for example. You can see here like water-soaked appearance on where the veins are, okay? So here are the examples of different symptoms. For example, in one type of lemon that I can't remember what was this in this case, the symptoms are over there, though much milder than what I've shown in the previous slides. Somehow here more. In sour orange, sometimes it's showing symptoms, sometimes not in the young flushes, okay? And mature leaves, sometimes this yellow vein appearances disappears, but some other like wavy appearance or water-soaked appearance of the leaves persist in the mature leaves, and very mild on some of the sweet oranges. As you can see here, it's almost invisible symptoms. Yet some more pictures of the yellow vein over here in Eureka lemon, sour orange. But one type of citrus, even though infected in the same stage of these plants, there is almost no symptom on it. So this virus can be symptomless. And here is the sweet orange that is very subtle symptoms on the leaves. I gathered some more symptoms over the other literatures and internet sources. So the pictures are not great, but still giving you some idea that the main symptom of this virus is causing the formation on the leaves and vein clearing or yellowing. Okay, here's interesting symptoms on satsumas. It's a really quite disturbing. As you can, hopefully you can see here, if you look closely, this slightly curled and backward twisted spring leaves showing some sort of infection going on. And some of the young leaves are back curled. And here it can be easily confused with the notching of ACP feeding, right? Remember ACP when it's feeding the young leaves, when that leaves matures, have this irregular shape or distorted leaf morphology, and we call it notching. So this virus also is causing similar symptom if you look at these leaves. And some of the satsumas, here is the healthy one, number 5, but others are producing out of shape fruits with the groove-like depressions, and they nicknamed it the pumpkin-like satsumas. So I'm assuming it will not be tasty and marketable fruit if this disease arrives to your satsumas. The most scary part, actually, this slide. All right, HLB has mainly one vector, is Asian citrus psyllid, but this virus can be transmitted with aphids, with whiteflies, with grafting, and with the equipment that you are using. So however you touch this virus, it is so easily transmitted to other plants. So that's why we have to be very, very careful and cautious not to bring this virus to our production areas. Okay? This is a systemic RNA virus that can be transmitted with number species of aphids and citrus whiteflies, and again, grafting and contaminated equipment and tools. So we have to definitely worry about this virus. And again, the same recommendation applies, same recommendation as leprosis applies for this virus too. The best management is not to bring this virus. And if you suspect any, symptom of this virus, please get in touch with us. We can help and stop the spread and, negative impact of this virus. So with that, I would like to stop here and acknowledge my team and my collaborators and all funding resources listed here. And nonetheless, I really thank again Mongi for organizing this seminar and taking care of the logistics, Alison and you in person who came and be part of today's seminar, and those who had joined us through the Zoom. Thank you very much for your attention. And if you have time, Mongi, I can take questions.
Thank you, Dr. Batuman. Any question from the audience? Any question from the Zoom? It looks like
I have one question, Ozgur. Thank you for the presentation.
Yes.
When you were speaking about Phytophthora, I wondered if there might be some, especially with respect to floods, possibly having increased the quantity of propagules that would be found in the soil. Is there anything that we can do in the way of drainage management that might help to reduce those propagules, or are we at risk as soon as we've experienced a flood and we'll need to remediate that propagule level, irrespective of what we do in terms of drainage?
Absolutely. Excellent point. Thanks for reminding me. It goes to show me that I skipped a point. I do remember writing, what if you flooded after Ian, right? So I really wanted to talk about that. As I mentioned, this pathogen is it's like a water mold. So wherever there's more water and the right temperature is the propagules or whatever the life form of the pathogen is present, multiplies and looks for energy sources, food. And what are the foods? Are fibrous, newly grown roots. So that's why it's very important that we have to drain our soil as soon as possible. One of the reasons is this, but other reason is citrus is not a water tree. So it doesn't like to have its roots in the soil. The water itself causes huge damages to citrus trees. So yes, we have to immediately drain the water after the flooding event and help trees to recover from not only hurricanes or flooding effect, but also elevated levels of propagules and the other pathogens in the soil. So our plants will recover much faster. And Megan, I think you wanted to add something?
Thank you. Yeah, I also wanted to point out that the flooding, depending on how deep the water was, it will also cause lesions up the trunk. So yes, Ozgur's absolutely correct. We want to get the water away as quickly as possible. If you did have flooding after Ian, hopefully you were either already on a Phytophthora program for root management, or you initiated one after verifying that you had enough propagules in the soil. So Ozgur did mention that those thresholds were established about 30 years ago, but Doctors Graham and Johnson did reevaluate those levels, because obviously there was the thought that maybe we needed to reassess them, but they didn't find any evidence that they needed to reassess them, despite the fact that we have this churn in the root system. And that churn is actually caused by a shortening of the lifespan of roots. So anything that you can do and we see that in the canopy too, right? So our leaves don't last as long, they fall earlier. The same thing's happening to the fibrous roots. They're dying much faster. So it produces just as many fibrous roots, but they don't last. And then the Phytophthora just takes advantage of that situation as they're stressed and they're dying. They're attracting Phytophthora by what they're giving out. So that's sort of part of what the dynamic is with the Phytophthora. So I just wanted to point out that, but I also am not sure, Ozgur, I know you gave the advice of possibly if you're putting on OTC, skipping that spring root flush treatment for Phytophthora. And I'm a little nervous about that. And that's because it is our major root flush of the year. And we want to keep every root, as far as I'm concerned, even, you know, Phytophthora was capable of taking out your root system prior to OTC and HLB. And I have a feeling it's just as capable of keeping that done. And we don't know what the oxytetracycline is going to do for the root systems yet. I think that remains to be seen as we get more experience with it. I really hope it does good things.
Excellent points. And the OTC idea was, I put just because to initiate this discussion. Thank you very much, Megan. So as you can see, we will all have different ideas, okay? So you have to think by yourself, looking at the existing evidence, what can be done or what was done and provided, you know, some effective control on diseases that we have, and then make the right decision. But this is definitely begs you to change some practices. For example, my wishful thinking that OTC, I don't care if it's gonna reduce the inoculum or not that much, but my hope is with a little bit of this inoculum suppression, maybe we'll give tree to sync again to give synchronized flush cycle or synchronized root cycle so that we can control the vectors or other diseases. Okay, that's what I'm hoping for. So, but this yet to be seen. Maybe this year will be the first time the OTC will be injected, and we will follow, of course, to see how plants are responding to this OTC. And again, I brought this example in the thinking or cloudy, cartoonish way to stir up the discussion. So this needs to be discussed, and this needs to be proven experimentally that what I'm suggesting is true or not. Okay. Thank you for bringing up, Megan.
I may have missed your earlier point, but where is leprosis currently located? Is it in Mexico or Brazil?
Both.
Both Mexico, Brazil, Colombia, Venezuela, Costa Rica, many Caribbean and South American countries. Unfortunately, leprosis present and causing some economic losses. And as we all very well know, that once you have a problem in your neighbors, that problem will soon become your problem too. So we have to be, that's why, very vigilant and not to bring any material, production material from other countries, at least that we can.
In Texas or other states in the U.S.?
No, no other states in U.S. There were some scare here and there, but their presence were not confirmed or verified. So no, there is no leprosis in United States for now.
Thank you.
Thank you, Dr. Batuman.